I know you'd like to have Irish coffee, but almost we are ready to present. Hopefully all young investigators are here. So just I'd like to make a brief announcement. Last time we had a Tarrytown 2 meeting with 17 young investigators, and most all of them in the field of ALS, and some of them really in upfront in research and so forth. And James Berry and Jinsey Andrews and Nazem Atassi and other people also doing other studies. So today we are going to have young investigators presentation. First of all, I'd like to ask Amanda Hydet Phillips from MDA. Would you stand up? She just joined recently MDA, and MDA, of course, NIH and MNDA and ALS Association all are funding this young investigators. So I ask Amelie and Lucie and also Bruijn to name young investigators. So if you are named, please stand and they are going to introduce. As you know, young investigators are our future, and do clinical trials, more appropriate, good clinical trials, the final answer to get final effective treatment. So I really appreciate your presentation. Amelie?
So we are absolutely delighted that we have 14 young clinical investigators at the workshop today, and I think all of them have been involved in the workshop preparations. And we actually had access to the bio sketches, and I have to say all of them have already very impressive accomplishments, and they are all currently involved in very exciting and cutting-edge clinical research projects in ALS. So Lucie, Bruijn, and I are going to take turns, and I'll start. So the first young clinical investigator whom I would like to introduce is Nathan Staff. So Nathan, please stand up. So Nathan is an assistant professor of neurology at the Mayo College of Medicine in Rochester. He received his MD PhD from the Feinberg School of Medicine at Northwestern, and he completed a neurology residency and 2 neuromuscular fellowships. And he also has a PhD in neurophysiology. And I also wanted to mention that Nathan has a KO8 award from the National Cancer Institute, and with that funding, he is looking at chemotherapy-induced peripheral neuropathy, and of course, that's also a very important research area. Next is Stephen Goatman. So Stephen is a clinical assistant professor at the Department of Neurology at the University of Michigan, and he's also the director of the ALS Association Certified ALS Clinic at the University of Michigan. So Stephen received his MD from the University of Chicago Pritzker School of Medicine, and he also holds a master's degree in clinical research design and statistical analysis, and of course that knowledge is essential for our workshop. And I also wanted to mention that Steve is a co-investigator on a CDC-funded study that is looking at environmental factors that contribute to ALS incidence and prevalence. And he's also involved in 2 interventional clinical trials for ALS. Next, I would like to introduce Senda Iro-Driss. So Senda is an associate professor of neurology at Northwestern. She's an academic neurologist certified in neuromuscular medicine, and Senda received her MD from the Medical School of Tunis. So Senda is being trained by in molecular genetics of ALS in Tipu Siddique's laboratory. So of course that is a world-leading research team for that type of research. And Senda also has an interest in imaging in ALS, and I heard that she recently participated in the NECELS imaging satellite meeting in Orlando. The next young investigator is Giorgio Manousakis. So Georgios is an assistant professor of neurology at the University of Minnesota. He received his MD from the University of Athens, and he did a neuromuscular medicine fellowship at Washington University. And I wanted to mention that because during that time, he was involved in the phase 1 clinical trial of an antisense oligonucleotide for SOD1-linked ALS. And of course, that program is still moving forward and is now being funded and conducted by Biogen. And next is Harry Bano. Harry, please stand up. So Harry is a visiting assistant professor at Harvard Medical School, and he's enrolled in a master's program in clinical and translational investigation at Harvard. He's also a neurodegeneration clinical research fellow at Mass General Hospital and Brigham and Women's, and he's also an assistant professor at Nagoya University Graduate School of Medicine. So Harry got his MD from the Nagoya University School of Medicine, and he also has a PhD in neuroscience. And I did want to mention that I've worked with Harry over the past couple of weeks on the preclinical study session, and Harry is going to give a presentation tomorrow.
So next is Catherine Nicholson. She has currently has a therapeutic development fellowship at MGH and Biogen, and is gaining experience in designing observational studies and conducting clinical trials at ALS at MGH. And she has recently contributed to a review evaluating past ALS clinical trial design and future innovative designs approaches in the Journal of Neurotherapeutics, so really relevant to the workshop as well. Thank you. Sabrina Pagnoni, who is she's a key member of the ALS research team at MGH and joined as a research fellow in 2012. Worked closely on a number of projects, including database analysis. Has personally been tremendous for the ALS Association as a judge for our Assistive Technology Challenge, and has been very involved in opportunities for new clinical trials coming on the horizon. She obtained funding both from NIH, a K Award, and several other foundations, and has really developed her independence and leadership in research activities, and a great team player. Thank you, Sabrina. Shafiq Lada, he received his medical degree. He received his medical degree from the University of Arizona College of Medicine, is very involved in multiple clinical trials, and has arrived in Barrow now since returned to Barrow in 2007, where he's the director of the MDA clinic and involved in many clinical trials, several funded by the ALS Association and other teams. Thank you, Shafiq. Amar Jadat. He is a practicing neurologist in Kansas. He graduated from the University of Baghdad and has been in practice for the last 13 years, and he is currently the director of the ALS Certified Center at Kansas. Thank you. And from Canada, we have Marvin Chum, and he is an ALS fellow, currently working with Dr. Shoesmith, and he has been funded by the ALS Canada for the last 2 years for a clinical training grant. And he is also doing his master's in clinical research, design, and statistical analysis at the University of Michigan, which he started in this last year and hopes to complete in 2017. So thank you very much.
Thank you, Lucie, and I'd also like to say a thank you to Hiroshi, because I thought it was a really inspired idea in bringing so many young investigators to this meeting. So we have a small crop from the UK. First of all, I'd like to introduce Dr. Rebecca Broad. Where are you, Rebecca? Rebecca's a clinical research fellow in motor neuron disease at the University of Brighton and Sussex Medical School, working with Nigel Leigh on transcranial magnetic stimulation and neuroimaging. I often talk about her work as being a bit of noddy science. NODI standing for, hang on a minute, Rebecca, I have to get this. Neurite, orientation, dispersion, and density imaging. I should have put my glasses on before I read this. I'll do that now. Thank you, Rebecca, and welcome to the meeting. I'd like to introduce Dr. Rebecca Belandra from University College London. Rebecca is currently a Wellcome Trust Clinical Research Fellow working at the Institute of Neurology at UCL with Ricky Patani and Adrian Isaacs. She works on iPS cell and Drosophila models of ALS, but she did want me to mention Amar Al-Chalabi, who she credits with really getting her engaged and enthused about ALS research, and indeed she did a lot of the clinical staging work with Amar. So well done, Amar. That just reminds me, I think I'm getting on a bit because I remember presenting a young investigator prize to Amar Al-Chalabi. And in those days, yeah, it was about, I don't know, how long ago was it, Amar? But yeah. I think we were both young then, weren't we? Happy days. Next fellow is Dr. Jonathan Cooper-Knock. And Jonathan's a clinical research fellow at the Sheffield Institute for Translational Neuroscience, where he works with Pam Shaw. I just don't have time to talk about the number of pies that Jonathan has fingers in, but he does a whole array of genetic, genomic, and cell biology studies. And last but certainly not least, Dr. Malcolm Proudfoot from the University of Oxford. And Malcolm is another Wellcome Trust Clinical Research Fellow, and his focus is incorporating magnetoencephalography and MR spectroscopy into the multimodal BioMOX longitudinal study, working very closely with Martin Turner and Kevin Talbot.
Thank you very much. Really appreciate it. Now, Bob and Nigel.
Switch on the microphone. Let me try it again. Isn't it incredibly exciting, the amount of young blood and energy coming into this field? For those of us who've been doing this for a very long time, it is so exciting to see that there are people who want to get into the field and still find ALS and neurodegenerative disease an exciting area, and it certainly is. So bravo, welcome, very, very exciting. Nigel Leigh is my co-chair, and he's going to take the first prize.
Okay, so we put up some ideas here, but that's by no means exclusive. It was just what we put together when we were asked by Hiroshi to try and have some concepts to get this going. I don't think we'll have any need to stimulate it because we have three extremely, probably overstimulating speakers. Certainly, the first one, Ettore Beghi, I'll introduce now. We were just reminiscing, as old friends do, and thinking we go back to the specialist trial. Who remembers the specialist trial? There, well, one. Is it all? Good memory. Yeah, I had to remind you. So this was a trial of branch chain amino acids, and actually we have come a bit of way since that time. Ettore is eminently qualified for this role of trying to translate or trying to stimulate our ideas about trials and broaden them, because he is a Renaissance man, of course, from Milano, where he's director of neurology at the Mario Negri Institute, but he's trained also at Rochester at the Mayo Clinic and at Monza. He is a man of many interests, as I say. He has done epidemiology and clinical trials across almost all areas of neurology. Maybe one or two that I've identified he hasn't been involved in, and seminally as well. And of course, he's been helping in design trials. So our brief was to ask our colleagues who are contributing tonight, not just to describe what they have learned from their pet areas of research, I think epilepsy, particularly for you, and that's what your brief was. But translating it that way, what are the lessons that we can learn? And we want to stimulate the discussion from that. So, Ettore, thank you very much.
Thank you very much, Nigel. Thank you very much to Hiroshi for inviting me. And thank you to you. I hope that you are still awake, but I take just 10 to 15 minutes for you. Okay, what I'm going to show you here is the disclaimer. And then I will address these 4 points. I will shortly mention the peculiarities of epilepsy, and then I will address the problems that we face with studies in epilepsy. I will compare epilepsy and ALS for similarities and differences, and I will conclude by looking how studies on epilepsy can inform randomized trials in ALS. What are the characteristics of epilepsy? Well, epilepsy is an heterogeneous clinical condition which is only in part reflected by having differing etiologies. Comorbidities are not rare. The disease is progressive or deadly just in rare cases, so this is a huge difference compared to ALS. We see that in about 50% of patients, seizures that are the symptom of epilepsy tend to abate, and the patient has no more relapses. The patterns in the outcome of the disease may be variable across patients. This is something that can be useful also for ALS. And seizures are the only measure that we have to evaluate treatment efficacy. However, in assessing treatments in epilepsy, we have problems. The first problem that we have is that all the drugs that we have in our hands are symptomatic. So they are only active on seizures and they have no impact on the progression of the disease. Another point that is very important and that should raise a discussion between us is that in epilepsy, polytherapy adds very little to monotherapy. So if a patient fails on monotherapy and you add another drug, the probability of a better outcome is very rare. So this is even present if we include in the regimen drugs that have different mechanisms of action. So even though we may address different points and there is a rationale to use different drugs, the end point is that you don't improve the outcome of epilepsy. Unfortunately, in epilepsy, there is almost nothing in terms of developing drugs that abort the epileptogenic process. This is something that happens at the presence of serious CNS insults in which patients may have seizures after a latency that has behind an increasing epileptogenicity that is the cause of the seizure themselves. Another point is that we almost never use severity scales, and this is something that unfortunately does not compare with ALS. And again, unfortunately, biomarkers are as yet unavailable. What similarities and what differences we have when we compare ALS and epilepsy? As I said, both clinical conditions are heterogeneous and both are associated with comorbidity, but in epilepsy they are sometimes overlooked, and I assume that in ALS they are frequently overlooked because the focus is on the disease itself, and maybe that the other clinical conditions are not followed with the same attention that is paid to the disease. In ALS, the only treatment that you know available is Riluzole, which is minimally effective, and you have a bunch of symptomatic drugs. In epilepsy, we have several effective drugs, but they are only symptomatic. Preclinical studies for epilepsy deal with a number of different animal models, while you know that in ALS we have just one animal model that is important. Phase 2 trials are similar because in general patients that enter phase 2 trials are drug-resistant epilepsy, so prevalent epilepsy people, chronic people that receive a treatment that is optimized, and on top of that a new drug is compared to placebo. And this is interesting. I would rise to another point that I would be very happy to discuss with you. And it came out this morning while discussing the other treatments that a patient with ALS is doing. Well, the attempt is made for epilepsy to use the best treatments and the best dosages in order to have in the background treatment of the patient the best available treatment. This means that if we could make a parallel to ALS, that they should receive the best symptomatic treatment in order to have any type of control by the available type of treatments and see if we improve even more the disease, this can be attributed to the drug itself. Phase 3 trials are characterized for epilepsy by add-on treatment, and then we have monotherapy treatments for newly diagnosed patients. And this is another important issue that I will address at the end of my presentation, because we in epilepsy have two types of trials. One is dedicated to chronic drug-resistant patients, and the other one is for newly diagnosed patients that obviously have a different clinical situation. The duration of the trials is between 6 and 8 months for ALS, and in general for add-on is 6 months of treatment, while for monotherapy, as the patient is less severe, we have a longer follow-up, is about 12 months. And both in ALS and in epilepsy, we have other non-pharmacological treatment that is mentioned here. What are the key points in the trials that may serve from the experience maturated for epilepsy for ALS? Well, we have drug-resistant epilepsy patients, and we enroll these patients at a time in which they have a minimum number of seizures in a short period of time. So if we want to make a parallel for ALS, we should identify patients with similar disability levels at the time of enrollment in order to have a fairly homogeneous sample. Newly diagnosed patients, patients with different seizure types, again for ALS, we could consider the possibility of trials in newly diagnosed patients. This issue came out early this morning. We have selected the epilepsy syndrome, so the heterogeneity of the disease is such that we have different types of epilepsies, and we have different types of trials in these different diseases. And the same could be considered for ALS. And then we have patients at high risk of seizures, which still, as I said, are the object of trials in its infancy, so we don't have suggestions for ALS, but a parallel can be made for healthy subjects that carry ALS pathogenic mutations and that you are seriously considered these type of trials. What about the outcome measures? Well, in drug-resistant epilepsy, we should reduce the seizures for a given amount, a given percent. So I wonder whether we could use given disability levels as an endpoint and say, well, if the patient does not achieve that worsening of the functional scales, maybe that the drug works. Or the patient becomes a failure at the time in which the scale arrives to a given score. Newly diagnosed patients, again, and this can be done also for ALS, looking at the time taken to arrive to a given score in the disability level. Then for epilepsy syndrome, again, we have the same as what I said before. And for patients at high risk of seizures, this is the time to arrive to the first unprovoked seizure. And for the same for ALS, we can consider eventually the possibility that the individual that is given treatment arrives to have the first symptom of ALS. Adverse events and quality of life are managed in the way as it's done for ALS, so there is nothing to suggest from trials in epilepsy. So this is the last slide. I would like to summarize some sort of take-home messages from the epilepsy trials. Patients with different phenotypes require separate study designs. So I wonder whether these can be considered also for ALS by separating newly diagnosed patients with chronic disease but not in terminal stage or end-stage ALS, which could be the object of different types of trials, or eventually, going back to the discussion on stratification, a good point for stratified patients. Treatment effects are based on substantial reduction of seizure frequency. Again, the treatment failure can be measured by meeting specific endpoints that represent a worsening of the patient. And in this specific point, we can use the word responder. So a patient is a responder provided that this patient does not meet a specific endpoint that is reaching a given score in a disability scale. And last, no, there is another one. A new drug should be non-inferior to the available treatment. This is what happens in epilepsy. And so I wonder whether any new drug should be compared to riluzole and considered at least non-inferior to riluzole. And finally, the presence of effective drugs makes placebo unacceptable, even in add-on trials. And there is a lot of discussion in the epilepsy field on this specific issue. So should we use riluzole as an active comparator instead of placebo? Thank you very much for your attention.
Would you like to stay there? We're planning to have some questions now and then obviously hold over for an open session after the other speakers. But if there are questions now, let's go. What?
Ettore, there are epileptic, anti-epileptic drugs that work on certain subtypes of epilepsy. Do you do comparisons with those drugs across the whole field of epileptic types, or should you concentrate on individual types? And that means in relation to riluzole.
Well, the large majority of people with epilepsy are treated with a group of drugs that are virtually the same in terms of efficacy. We have selected types of epilepsy, especially in small children, in which you use specific drugs that are not used in other epilepsies, but these are rare instances. So in the majority of the patients, we have a lot of drugs that are virtually the same in terms of efficacy. For this reason, we generally make our choice based on the safety and tolerability profile of the drug.
Thank you. Ben.
You reopened the responder issue again, and thank you very much. You did a trial with acetyl L-carnitine where you specified that the patients had to not have gone past a certain point. And how do you think we should analyze these responders? By a functional scale? You know, I mean,
I think that the functional scale would be the best way, because, you know, you should use something that is sufficiently sensitive to pick up changes, and the patient is not in the end stage, so we have to use survival in that case. So, for example, I thank you for mentioning that trial, because we identified 3 specific elements in the ALS scale that represented 3 important functions in the daily life of a patient, and we said that at the time in which a patient failed in one of those points by accumulating 3 or more in the score, the patient became a failure to the treatment. And this could be interesting. Actually, that type of modification of the scale was validated and has been recently published in ALS.
In that context, then, should we be doing more of those trials? I mean, particularly, I mean, it sounds like the edaravone story is, that was a population you chose, a very early population,
Yep.
and you were able to have a positive outcome.
One of the striking things about epilepsy science is that with the discovery of genetic mutations in particular channels, as far as I can see, that's produced absolutely no incremental improvement in therapy. Do you think that has any lessons for ALS?
No. Actually, there are drugs that have been discovered, and again, looking for drugs that act on these channels. There are drugs that act on the potassium channel, other on calcium, other on sodium, and if you measure their effectiveness on clinical grounds, you don't see any difference.
So, I mean, I think the point really I'm getting at is that epilepsy drugs arose through phenotype-driven screens, so, you know, slice cultures where you had kindling and various mechanisms, and you know, it makes me wonder whether through understanding very basic molecular interactions, which I would normally propose as the right way to proceed, you know, we should go like that, or through phenotype screens where you actually have to see some disease-relevant mechanism in a cell culture.
Yes, but unfortunately, I don't have an answer for your question, because even though you may see in preclinical studies that there are effects on specific items that refer to in vitro studies or to experimental animals, when you go to the clinic, you don't see anything that helps in making differentiations and comparisons across drugs. There is, unfortunately, there is no way. As a matter of fact, the epilepsy field is moving to very different targets. One is inflammation, for example, to see if there is the possibility really to approach the process at the time in which we are in the early phase, in order to abort what is behind the seizures, that is something that maintains unprovoked seizures. But we are still very, very, very behind schedule.
Is there another question then? Okay. We'll come back to you. Our next speaker hails from the University of Rochester, where he is the Robert Joint Professor of Neurology, Senior Associate Dean for Clinical Research, and Director of the Clinical and Translational Science Institute at the University of Rochester. Karl Kieburtz is a Professor of Public Health Science and of Environmental Medicine, and he was the founding director of the Center for Human Experimental Therapeutics at the University of Rochester. His primary clinical and research interests are neurodegenerative diseases, and he's been very active in Parkinson's disease, Huntington's disease, and HIV-related disorders. He is the PI for the NIMDS-sponsored trial of neuroprotective agents for PD, and he directed the Coordination Center for an NEI-funded consortium in neuroophthalmology. He did all of his training at the University of Rochester, which is a center for clinical trials in neurologic disease with very few peers and equals. We really appreciate you coming here at college to share lessons learned from clinical trials in other neurodegenerative diseases across the Americas.
Thanks, Bob, and thanks to Hiroshi for inviting me and for the chairs for guiding me. I'm going to talk primarily about the UPDRS, the Unified Parkinson's Disease Rating Scale, and hopefully it'll have some analogies to the ALS-FRS-R, at least as one kind of bridge between Parkinson's disease and ALS. I'm going to talk about three main things about the UPDRS. One is the vagaries of how and when you measure it. Two is how it might relate to measures of disease activity and how it can fall down there. 3, how it relates to patient experience and how it can fall down there, yet it is still the primary outcome measure for most studies that are used to approve drugs in Parkinson's disease. And then talk a little bit about potential solutions from rocket science. So Parkinson's disease, most of you know, is progressive neurodegenerative disorder. It has a neuropathologic diagnosis based on loss of cells in the substantia nigra, but there's other diffuse pathology. Diagnosis is made on clinical observation. Signs and symptoms are primarily motor, bradykinesia, akinesia, rigidity, tremor at rest, eventually postural instability and gait impairment. But there's a lot of non-motor features also, cognitive impairment, and a host of other rather non-specific features, but clearly are part of the Parkinsonism syndrome. For those of you in the back, you can probably see the color, but you can't see much else. This is just the idea that at least in PD there is a identifiable prodromal period, which can be characterized by some non-specific things such as constipation, REM behavior, sleep disorder, although the latter one is not so non-specific. But at some point a diagnosis is made. Those non-motor features are represented by this quadrant. Then motor features develop over time, and then there are so-called complications that develop over time, psychosis, motor fluctuations, dyskinesias, other things which are probably plastic responses to the dopaminergic treatments we have. And we kind of divide that into early and late disease depending on whether things are complicated or relatively straightforward and still primarily focused on motor. That said, there's a tremendous variability in clinical progression. And there's this broad spectrum of symptoms leads to various ways of thinking about subdividing groups in Parkinson's disease. So age of onset is one way of thinking about that, early versus late onset. Early onset is particularly associated with more rapid development of motor complications. So if you think Michael J. Fox onset in the 4th or 5th decade, people live quite a long time with that, but for whatever reason, priming with levodopa seems to elicit motor complications much more quickly and to a greater degree in terms of their severity than older onset patients. That's why most of the young onset patients go on to DBS, from which they can get quite a good response. So that's one. Age at onset is one form of heterogeneity. Motor phenotype is another one. Tremor at rest, asymmetrical rest tremor, one of the key hallmarks of Parkinson's disease, but absent in a third of the patients who carry the diagnosis. So, and the rate of progression of the tremor predominant versus the so-called akinetic rigid form, quite different, with people with tremor predominance progressing more slowly. People can just be categorized as fast or slow progressors. About 15% of patients over a 5-year period will have minimal to no progression of Parkinsonian signs and symptoms, which either suggests they're a very slow progressor or they have a different disease or perhaps no disease at all. A little more about that in a minute. And then cognitive impairment. It seems that there is everyone has some degree of cognitive impairment with Parkinson's disease, but there seems to be a subset that's at greater risk of progressing to Parkinson's disease dementia. And then, of course, there's genetic variants with the leucine-rich region kinase defects, as well as in the GBA, same gene defect as homozygous leads to Gaucher's disease. Heterozygotes are at increased risk of Parkinson's disease, and they appear to have a faster rate of progression on cognition, at least, and the LRRK2 carriers perhaps on motor features, although that remains open. So just to say, there's a lot of ways of sub-segmenting the population in ways that the clinical phenotype seems to progress differently. That said, we tend to use one measure. It's called the UPDRS. Part 1, this is the traditional one. There's a revised version called the Movement Disorder Society UPDRS, but it's not terribly different. So part 1 is about non-motor features. Part 2 is about activities of daily living. Part 3 is about motor function. These are scored 0 to 4. It's not there. It's mild, moderate, severe. It's terrible. That's sort of 0 to 4. And we add that up and we call that a continuous scale. So it's really just an ordinal, 44 ordinal items which are added up to act as a pseudo-continuous measure. All the drugs that are approved for Parkinson's disease and early Parkinson's disease and to a certain extent later Parkinson's disease rely on this outcome measure. So how does that behave over time? So this is a graph. Down here is the points on the scale. It goes up to in the high 100s, but practically speaking, it has an operational range between 0 and about 80. This is the duration of the disease. This goes up to 40 here. This is the duration of the disease out to 15. And what I want you to see here in red are early untreated Parkinson's disease. That's the rate of progression over that period of observation in the red score. So what you can see in early untreated PD, this is diagnosed but not yet on any treatment, they progress at a slope of about 8 points a year. Turns out in Parkinson's disease, unlike other neurodegenerative diseases, we actually have very potent treatments. And at some point, people go on those treatments. And these green lines reflect the rate of UPDRS progression when you're treated. So they're later in the disease process, because treatment tends to come later. And you'll also see that this UPDRS rate of decline falls by about a factor of 4. The other thing you can see here, perhaps, in this intersection of these two lines, if you're close enough, is if the absolute score you start at seems to predict the rate at which you progress. So this group starts at a higher score and they go faster than this group that started at a lower score and goes slower. So you can see how you define people, early versus late, more or less severe will determine the rate of change in your outcome measure. This is just a publication in the New England Journal. I'll just say that this tells you that all these people came in with a UPDRS score of about 30, and here's the placebo arm showing you over 9 months. Stan Fon was the first author on this. People progress about 8 points over 9 months, so about 10 points over a year. This is the first dosage ranging study of levodopa. It was amazing that we didn't do this for 30 years with one of the most potent drugs in neurology. But this also just shows that after washout, people who were treated did better than people who were not treated. So this suggests that there was something about early levodopa treatment that was not, that was enduring after washout, and that these people did better. So on their UPDRS scores, those people did better than the people on placebo. You won't be able to see these scores, but there's two things I want to say. So this is a measure of actual disease measure. This is beta CIT imaging of dopamine reuptake. So thought to be a way of imaging the severity of disease progression. Two things here. One, about 15% of the people who entered the trial with the best Parkinson's disease experts in the country, theoretically, 15% of them didn't have the right diagnosis based on this. And then Chris Coffey's somewhere in the room. He can tell us that in the relatively recent PPMI trial, it's about the same. So if you do dopamine transporter imaging, you find that about 15% of people are clinically misdiagnosed. And if you do that in this study, the rate of progression on that measure of disease activity is actually higher in the groups that did better clinically. So how come the score of the clinical outcome is divergent from the underlying disease progression measure? I don't know, but that's just the way it is. And in 50% of trials where such imaging is done, the clinical benefit on UPDRS is divergent from the underlying disease measure. This, I'll just say, tells you that these patients are people who are randomized. This is in JAMA and JAMA Neurology, looking at initiating 2 different treatments in Parkinson's disease. Their baseline UPDRS score was just about the same as the last one I showed. And there's a curve that looks about the same. The 2 things here, note that at least we have very potent symptomatic treatments. This is starting at about 30 and driving down by 30% to 50% improvement in that disease severity score. And this curve looks very similar to that previous placebo curve we saw in the levodopa study. The only problem is the time frame, instead of being 9 months, is 4 years. So this just points out that the rate of progression is slowed very substantially by the presence of treatment. So let me just point out the levodopa curve here. This is the more profound benefit, and you can see that patients 4 years after initiating levodopa are still not back to the level of severity when they entered the study. That's on their UPDRS score. This is their quality of life measure. So here's levodopa in this pink line. Yes, when you start levodopa, their quality of life improves. This is the Euroqual It's not the EQ5D, but the visual analog scale, but they showed basically the same thing. So anyway, people get better, but by about 2 years, their quality of life is back to where they were at baseline. And by 4 years, their quality of life is as much deteriorated as it was ever improved by Cinemet. But when you look back, if you were to look at their UPDRS score, you would say they're still better than they were at baseline. So this just goes to show that the physician-guided rating of disease severity does not align with the patient-reported quality of life. So what are we supposed to do about this? Well, UPDRS is a measure we're going to use in clinical trials. How can we optimize using it? Modeling and simulation might be a way to help us in that. Modeling and simulation is something which is old to the aerospace industry. No one put up a space shuttle before there was a lot of modeling and simulation, including things like atmospheric, wind speed. Basic question was, how much weight can you get into orbit? And that was, many potential ideas were tried and failed quickly before ever trying to actually launch a space shuttle. And of course, modeling and simulation is active in other diseases, in HIV/AIDS, type 2 diabetes, non-small cell lung carcinoma, especially about tumor growth models and survival, although the next speaker may have more to say about that. But certainly about HIV and diabetes, a lot of modeling is done. But we've done very little modeling in neurodegenerative disease, except there is recently published an Alzheimer's disease model. I'll just say quickly about it. It used both the natural history progression, effects of intervention, and also simulating potential clinical trial designs. 5 things went into this model. I'll just move through them very quickly. There's an equation for you for late at night. Won't talk about that, and please don't ask me a question about that. Factored in also is a so-called Bateman function, which looks at transient placebo effects. Also factored into the model are the background treatments, like this is donepezil versus placebo. It actually changes the trajectory over time and dampens the curve of progression. And this is looking at covariates that can influence the severity of cognition. So this is the ADAS-Cog, again, similar to ALS-FRS, but looking at the baseline severity of cognitive impairment using a slightly orthogonal measure, the mini-mental state. So here you can see if you start with a mini-mental state at 26, your progression of ADAS-Cog is very slow. If you go down to 21, it's very quick. So your baseline severity of the parameter of interest, cognition, is highly predictive of the rate of progression. You could imagine, I don't know, forced vital capacity being a predictor of how quickly the ALS-FRS projects. And this is looking at another completely different phenomena, but using the same thing, mini-mental state, as a predictor of dropout. So the proportion that will drop out, the lower the mini-mental state, the more likely you are to drop out. Those 5 things amalgamated together allow you to make a predictive model on progression of ADAS-Cog scores. Same kind of thing can be done in Parkinson's disease. This is the UPDRS. We've created such models using both an aggressive early deterioration and impact of symptomatic treatment, and then progression over time to an asymptotic behavior. And where you can't see these in the back, but using mathematical models abstracted from other data sets, we're able to then, in retrospect, apply them to existing clinical trial data sets. And using those relatively simple models, you can predict the outcomes in clinical trials within about 10%, using just baseline characteristics. So this suggests that using modeling might be a way of optimizing, projecting what outcome measure behavior is going to be like, so that we can think about sample sizes, entry criteria regarding severity of baseline illness, concomitant medications, to get a better handle on what we might expect in terms of outcome measures at the end of a trial. I'll stop there. Thanks.
Paper is open to questions. I'll ask one while people are coming to the microphone. The idea of modeling is something that has just begun to appear for us also. Are you talking about using the model to predict the outcome of, say, the placebo group, and then look at the other group as a comparative, the treatment group? Or are you also trying to model the prediction of the response to the drug?
trying to predict what you might expect from a placebo group given a certain set of entry criteria that are, relevant in the model, and then using whatever the projected, for example, treatment effect size you have of interest, 20% reduction, 30% reduction, which is not necessarily database, but maybe just aspirational, to then try to get study parameters. How many people do you need? What's the impact of dropping the baseline severity from X to X over 2? What does that do to sample size? What does that do to duration to be able to see those differences?
And is the FDA open to this kind of?
So that model in Alzheimer's disease has been accepted as fit for purpose by both the FDA and the EMA, not for approving drugs, but as a tool which will be useful to academia and industry for planning for clinical trials.
Yeah, I'm Mike McGrath, University of California, San Francisco, and I'm happy that there's a session like this talking about what other diseases can tell us about what we should be doing. And I'm also happy you brought up the whole AIDS issue. One of the nice things about Parkinson's is that it's really systemic, right? In other words, you can get access to gut, ganglia, or whatever else, biopsies. Because one of the things that I don't know if we talk about enough, is how can you prove that you actually have a disease-modifying drug that actually hits what's going on? It's hard to do spinal biopsies or brain biopsies, but with Parkinson's, I think we have a chance to be able to look in the gut and look at sort of pathogenic processes related to the dopaminergic neurons that are there. So that's one thing. Second thing is, you probably all know or don't know that AIDS has taken a huge turn in the last couple of months. It turns out that the antiretroviral drugs don't penetrate the tissues, and that the HIV is evolving in the tissue at the same rate as in untreated patients. And so we have a two-compartment disease. What a lesson, I tell you. I remember, I'm at San Francisco General for the last 30 years, and so I know a little bit about AIDS. And so we can cure one compartment, which is the blood, but the question is, what's going on in the tissue? And so that's what's attractive to me about Parkinson's, is the possibility of being able to look at an accessible site of tissue in the gut, because obviously we can't, as we're trying to figure out how to develop disease-modifying therapies, we need to be able to say, did I really do what I wanted to do, right? Did I actually change the pathophysiology of what's going on? And so right now, what we do is we just treat and pray a little bit, right? So anyway, what are your thoughts about being able to follow patients with Parkinson's using a peripheral source of tissue?
So people are interested in that. People have looked at colonic biopsies retrospectively and show that you can see alpha-synuclein deposition. You can also see Meissner and Auerbach layer degeneration early on. But that's kind of tough tissue to get at. People have also looked at salivary gland biopsies. Right now it looks a little too non-specific as a primary outcome measure. The other thing is looking at, with OCT, ocular coherence tomography, on retinal ganglion cell layers, but it's tough. The compartment of interest is hard to get at.
Yeah.
And just like HIV is protected in certain lymph nodes and in the brain, it could be that we're not getting our interventions to the compartment of interest for neurodegeneration. Very tricky to know.
So you don't think that I should be advocating doing Parkinson's studies and doing gut biopsies and follow them that way? I'm not that I've ever done that, but I'm just looking for ways of saying, I think that I can do this to the nerve system, whatever. And the question is, how can one prove that, at a histopathological level?
It is attractive to be able to do that, but it does beg the question, even if it's doing it in the gut, is it doing it in the brain? But at least you're biologically active on the disease process of interest. If you can see.
That's interesting. Thank you.
Thank you very much. And from the viewpoint of disease-modifying therapy, prodromal phase, preclinical phase, is very important. It's a target of the disease-modifying therapy. And as you know, in the case of an AD, Alzheimer's disease or Parkinson's disease, nowadays they are focusing on the prodromal phase, and by, for example, using a PET scan on the tau deposit or A-beta deposit. How about the ALS? What is a prodromal phase? And can we detect the prodromal phase by using, for example, TDP-43 PET scan or those kind of things? How do you think about that?
Well, it is an area that's tricky. Careful what you wish for in terms of prodromal investigation, because first of all, and there's FDA guidance about prodromal development of drugs, at least in the Alzheimer's world, that Rusty Katz and Nick Kozauer wrote a couple of years ago, which at least gives a framework for how to approach this. It is tricky because you have to be able to reliably identify that at-risk population, because you're going to expose them potentially. Yeah, and those agents have to be pretty well tolerated because you're intervening in people who are well, yet face perhaps a very bad outcome in the near future. People are bad at outcome estimation. That's why our lottery is so good, as we make a lot of money, because people are bad at probabilities. So their judgment and understanding those things is tricky, but it does present at least what seems to be the greatest therapeutic hope in neurodegeneration, is if you can identify people reliably and intervene at a mechanism that's responsible, you have the chance of completely forestalling illness, even though people have the disease. So I think that is a big 21st century challenge. And you know, there are trials ongoing in so-called pre-AD in the autosomal dominant AD world, and also in Huntington's disease, where we have deterministic genes, and I guess could be done in FALS also. And it gives an opportunity to make those kind of interventions. But it's a whole new world for us in how to do those and the risks and issues that go with it. But I think it's certainly a great opportunity.
And the last question.
Let's get down and dirty. This man here has a company that has looking at a model, and you've whetted our appetite here with your model, and there are models for different diseases. The Europeans have, and others have other models. How can we convince the FDA that we can use these models in analysis plans?
Yeah, no, I think fit for purpose for trial planning is one, but the ultimate aim would be that you have models that are sufficiently robust that your outcome measures are perturbations of the parameters of the model, rather than looking at the delta or some kind of MMRM or something of the ALS FRSR. That's the goal, because that should have more precision, because if those parameters are robust and you change them, that means the drug works. I think within the Office of Clinical Pharmacology, they're very interested in that. When you get out to the review divisions, they're not so keen on that. I think what drives that are fora like this, where there's a chance for industry, academia, and regulators to get together and advance that regulatory science. I think that's, not only is that important, I think it's essential, because it will drive us towards greater efficiency, and it's too inefficient the way we're developing drugs in these diseases now. And so I think that's part of the challenge, is getting that done.
Our next speaker is Dr. Andrew Vickers, who's a biostatistician, hails from Memorial Sloan Kettering in New York. His research falls into three broad areas: randomized trials, surgical outcomes research, and molecular marker studies. One of his books is entitled, What is a P-Value Anyway? One of his most recent papers begins with the sentence, "The U.S. clinical trial system is broken." The title of the paper is Clinical Trials, 4 Methodologic Fixes. It's a pleasure to welcome Dr. Andrew.
Yeah. Thank you very much for inviting me. I actually rejected this invitation about 4 times. And then they said, we'll speak at 8 o'clock at night after everyone's had a whiskey. And I'm like, okay, you sold me. So what am I going to talk about? Why are trials failing? How do you stop them failing is by doing trials that are actually friendly to patients. And the only way of doing that is what I call clinically integrated trials. And then finally the idea that we have to think differently about how we're doing trials. You do your trial over there, and you do your trial, all lots of individual trials working independently, rather than actually having one master trial running all at the same time for the benefit of all patients and our ability to look at several agents at the same time. Okay, so this was our first conversation. I want to be just like you. And I'm like, well, you really want to be joining a failed clinical trial enterprise, because this is well documented. Even in the New York Times, this isn't an esoteric debate amongst statisticians. This is in the editorial pages of the New York Times, that cancer trials are failing. How are they failing? Well, 2 years, 700 steps to get something approved. A step might be, writing to the IRB and then responding to their comments would be, 3 steps or something. 700 steps to get a trial approved. I mean, it's just absurd. What if you gave a party and no one came, right? 1 in 4 trials. Okay, I'm not saying, we didn't accrue fast enough or we didn't complete the trials. No one turned up at all. Not a single patient accrued. This is what you want in ALS, right? Okay, so this is a slide I've showed several times. Somebody from Pharma said, "Oh, that's not bad. Our latest trial was 30,000 patients, $30,000 per patient. It was $200 million to do 2 trials. So we had to make the trial really expensive, and now no one can afford it." I'm like, okay, well that works out, doesn't it? Why are they so expensive is we're spending fortunes making sure that we did the trial correctly. About 1/3 of the money. Now, why is that? Well, put something like you're doing a prostate cancer trial, and one of the inclusion criteria is histologically proven adenocarcinoma of the prostate. Okay, if they didn't have that, why are they having a radical prostatectomy? But anyway, you put it down there. So to audit that takes as long as it does to document that in the first place, and it's obviously completely unnecessary. So as a result of this, about half of US trials are conducted abroad, and I find that appalling and highly problematic. I have no reason to believe that the response to chemotherapy and the type of cancers you get in countries such as India are really going to be comparable to what we see in the US. So a lot of what I've been talking about is regulatory problems. I think a lot of the problems we have in clinical trials are self-imposed. This is the eligibility criteria. This is what I call the Microsoft Word effect, because people just cut and paste the protocols, and then they add a couple of more inclusion-exclusion criteria, and they cut and paste more. It's like junk DNA. So we're getting now, 100 eligibility criteria in trials. And this has very real effects. It's not only increasing our costs because of all this audit we have to do to make sure that everyone met all of those criteria, but it actually has real effects. So this is on the y-axis, you have the proportion of patients you're excluding. You want patients on trials. This is a big problem. We need more patients on trials. We're excluding all of them, and we're excluding more and more over time as we add more and more eligibility criteria, which is, kind of obvious. So this is an estimate I've seen in cancer, and I happened to, in my background reading, somebody said this in the ALS research too. Trial complexity is increasing 10% a year. So someone said, in 1999 when we did this, so that was 16 years ago. So in 16 years' time, if trial complexity is increasing at 10% a year, trials are going to be 5 times more complex, 5 times more expensive. We're going to be doing 80% fewer trials in 15 years than we do now, unless we do something about the fact we're making our trials overly complex. So I was talking to someone, before the meeting, and they said, participant burden, that might be an issue in cancer. It's not an issue in ALS. And I laughed, because every investigator I have ever spoken to, and I've made some warning about a trial, says, no, of course that's true in general, but it's very different in my particular field. Okay? And what do people say? ALS studies, slower than expected accrual. Only 10% of ALS study patients are going on trials. and you're getting 1 or 2 patients per site per month going on a trial. Now 10%, in cancer it's about 4 or 5% of adult cancer patients go on study, which is a disaster. 10% is a bigger disaster because we have many more cancer patients than you have ALS patients. So I miss a patient on trial, I have another 1,000 down the line. You don't. Okay, so this is not just a problem in cancer, it's a problem in ALS. So what is the problem, right? The problem is you have to sell a patient going on trial. So have I got a deal for you? Okay, come in. Normally you'd come in every 3 months, but now you're going to have to drive into Manhattan and find parking every month. We're going to give you a whole bunch of boring, uncomfortable, and time-consuming tests. Not only that, we're going to make you fill in questionnaires. But just so you understand, you not only have to fill them in a paper, I'm going to call you at home and hassle you and get you over the phone. One of the nurses will call you up. Now, best of all, the best thing is, you might be on placebo. Right? This is not a good used car salesman. All right, so how do we make trials patient-friendly so that patients will go on them? And it's an idea that with Professor Peter Scardino, who's the chair of the Department of Medicine of Surgery at Memorial Sloan Kettering, we call it the clinically integrated trial. And the definition of a clinically integrated trial is that the experience of the patient and the doctor is virtually indistinguishable whether or not the patient is randomized. Now consider that if you put a patient on trial, what happens to them is very separate. It's different. So, oh, that's the study patient. Send them to the MRI, give them, instead of doing the lung function, I'll do the lung function. I've got to get someone special in, and they're going to repeat it 3 times. And that's why patients don't like being on trials, because of this additional burden. We're going to say, you want to be able to say to a patient, if you go on this trial, okay, you might get a different treatment, but everything else is going to be the same. We're not going to hassle you. We're not going to make anything more difficult for you. And so the only way of doing that is to get data from routine practice. So I'll tell you, about some trials that I'm doing at MSK. The blood tests that we do to test for, I want to know after our trial whether the patient has recurred. Okay? So we need to take a blood test. Well, you're going to take that blood test as part of routine practice, right? Cancer doctors follow their patients to see if they've recurred. Questionnaires, you give questionnaires. Don't you want to know how your patients are doing anyway? Why not give them a questionnaire? Use that in your clinical consultation to help advise the patient. You can also use that for research. The same thing as my understanding in your field, you do these lung function tests. That can be part of routine care, can inform your clinical care of the patient, and can be used for research. And I think there's a general principle here. And I can say this to anyone in cancer, there's not much I want to know as a researcher that you shouldn't want to know as a doctor. So I'm conducting a trial right now, and one of the endpoints is hernia. Do you get hernia at the port site for a minimally invasive surgery? If the patient has a hernia, you need to know that, right? Because you've got to go and treat the patient. And I want to know that for my trial. So let's get all these data. If we can get the data from routine clinical practice, that can be the endpoint of our trial. Now, whenever I raise the point of doing these sorts of trials in routine clinical practice, people say, oh, well, they're imprecise. And we add all these things into our trials to make them more precise. We measure several times. We have long, complicated inclusion and exclusion criteria. Right? We have, we make all these checks and balances and procedures. And why do we put them in? Is because they make the trial more precise. Now, this is just a theoretical model. It isn't real data. But as we make a trial more precise here, we increase the power, we decrease noise, we improve precision. And that's why we put it in the trial, in the trial protocol. Okay? So that's all great, but you ignore the fact that there's a downside, right? There's no free lunch. What happens when you make trials more complicated, you add more procedures, is that it's more difficult to get the patient on the trial because you've got more exclusion criteria, you're asking the patient to do more stuff, or they drop out halfway through the trial. And so your patient accrual goes down. And so your actual power, taking into account the fact that, well, yes, you're increasing precision, but you're losing patients, that also goes down. And so by making trials more and more complicated, on paper you're making them better, but in fact you're making them a lot worse. And then there's the issue of the placebo, right? When we had a conversation, you said, what's the number 1 reason patients don't want to go on trials? And you said placebo. So I'm like, well, then don't design trials with placebo. In fact, I wrote a paper many years ago called Why Have Placebos in Clinical Trials? And we wrote that paper because we were asking people and no one could tell us. The reason to put a placebo is to control for bias in assessment of outcome. There's lots of other ways of doing that. And to control for the placebo effect. I really don't think that's such a big issue in your field. I think you could probably either ignore it completely or just use active controlled trials. If it's probably better to have a placebo, but not if you're losing lots and lots of patients. So I just want to tell you about the trial that I'm a PI of at MSK. We have 1,400 patients on the trial so far. We're accruing about 300 a year. About 60% of the patients are going on the study. Does anyone want to give me an estimate of how much this trial has cost so far? Or how much money we brought in for this trial? But typically this would cost on average $10 million. So I want to know how much it's cost us so far? Nothing. We have not sought any funding whatsoever for the trial. The only cost we incur in this trial basically is consent, which is taken at the same time as other consents like for using tissue or surgery. You can do these incredibly cheap trials if you integrate them into routine clinical practice. Very, very high acceptance. Really no problems with the patients at all. Okay, now the idea of multi-arm trials. So what do we currently do? Now I don't actually, I've read a few protocols, but let's just assume that this could happen. You've got 4 or 5 new agents that you're interested in, and you want to compare them with the current standard of care. So you conduct 4 separate trials. One's going on in Australia, there's one in Europe, there's one in the northeast of America, there's one in an institution. And they're all run separately, all with different protocols. They all have to be separately approved, and the results are evaluated entirely independently. So why not do this? Why not have one trial? There's one protocol that you get centrally approved, and you can randomize patients between all 5 arms. And because you're not repeating over and over again the same control arm, you can actually get more patients on. With the same number of patients, you can get more statistical power. And then there's something else that you can do, which is called adaptive randomization. So this is how it works. You've got a trial with one standard care and 4 new drugs. And you start off, so there's 5 arms. So you start off with a 20% probability of receiving any one of the 5 drugs. Well, the trial goes on. About a quarter of the way through the trial, you've got some idea of the effectiveness of these drugs. And so one of the drugs patients don't seem to be responding to, so you actually bias the coin. You make it less likely that people will be allocated to that particular drug. This drug is doing well. You make it more likely that the patient is being allocated to that drug. You carry on running the trial. Response rates are terrible for this drug. Close that arm. Open up another one. Right? You amend your master protocol. You have one master protocol but with rolling comparisons, opening and closing as you go along. Now, it's not just good for patients. This has actually been shown to have good statistical properties, that you actually find out what are the good drugs much quicker than running separate studies. So what's the deal for the patient now? What can you say to the patient? You can say, we're always doing research. If you take part in our research, it's no additional burden. I'm not really going to ask anything more from you than the clinical care, than the sort of things I would need in order to take care for you as a doctor. I'm not going to ask you to come in or do a test or anything like that. Now, because of this trial we're running, I don't know really what's the best drug, but the way the trials run, you have a higher than average chance of getting the best drug, right? If you go on this trial. Lots of new drugs. We're assessing how they work. We're going to give you the better chance of getting the best one. And most of our patients go on study. All right, now why am I saying that? Well, there's an interesting thing that I read in your sort of background material. We need to get more patients on trials. So how are we going to do it? Well, education. we need to educate patients about how important trials are. We're going to get them on our trial planning committees, and we're going to advertise, which all seems very, very sensible, but is not actually based on any grounded theory of what makes anybody change their behavior. And the field of study, what makes people change their behavior, is called behavioral economics. And saying most patients go on study is something called social proof, and is a validated way of making people change their behavior. I can tell you something, in my field, prostate cancer, the biggest problem is getting people with low-risk cancer to go on active surveillance rather than active treatment. And we normally have about a 50% success rate. One of the junior faculty started using behavioral economics negotiation theory in his clinical consultations. Now his rates of acceptance are 97%. These are not, and he's not a used car salesman, he's a bit of a shy guy actually. If you just use established principles of getting people to change their behavior, and you pay attention to the words you're using to patients and the way you present things to patients, you will get more patients on trial. So look, in conclusion, you know, it's all about perspiration, not inspiration. In clinical trials, it's 90% is logistics. It's only 10% the statistics. So, you know, I guess when you first spoke to me, you thought I was going to put in trial designs and so on and so forth. And yeah, we can do that, but we can't do that unless we get patients on trials. And that's really a matter of designing trials so that the logistics work out. Thank you very much.
Two speakers over here, one here. We can bring them all together. Can I ask the unthinkable question?
Yes.
Statement. Hiroshi, it sounds from what Dr. Vickers has said that this meeting is the problem. Here we are, making life more and more complex. This is the question I'm asking to all of us. We the problem here? And if so,
yes. Well, how are you making it more complex? I would ask that.
Well, you sat through endless discussions about nuancing many different aspects of tests that we might do, we might include, and you may have looked through the previously published guidelines. They're quite complex. Many alternatives. What you're giving us is a very simple, pared-down approach.
Yes.
Not the only one, no doubt, but an approach. So my question is, are we creating the problem, not resolving it? Sorry.
Well, okay. Sometimes you look in cancer. You sometimes do need complex trials. Maybe want something on MRI, you know, and then you can also, what you can do is you can do trials where you actually have separate consents, and you say, look, this is a very easy trial. You'll get the best drug. You don't have to get any more procedures. Most of our patients go on the trial. Patients will go on that. Like, all right, now you're on that trial. I'm wondering whether you would do something altruistic and help us, and also get a spinal tap or an MRI or something. Separate those two trials. Separate those two consents. Trials can be, you can make the design complex, you can do whatever you want, but you've got to bear in mind what you're going to sell to the patient. How are you going to get the patient on trial? Because unless you get the patient on trial, you know, the perfect trial that no one goes on is not really telling you very much scientifically.
Thank you very much. So we open it to questions.
Yeah. Can I ask one question? I assume a lot of clinical trials in cancer is also very complex.
Yes.
So let me ask a question.
That's the entire problem. That's what I'm fighting against.
Right. So
don't follow us.
What you said is just incredibly important. Are you a minority or you're a majority in cancer trials?
Well, you have sort of majority, average, minority. What's sort of below that? I'm like totally left field, right? In cancer, but you know, there is a growing number. There are a growing number of people. I mean, there's the City Initiative, there's the Sensible Guidelines. There is, you know, what I wrote in clinical trials. I wrote, you know, you read a paper, and you'll notice that 4 of the top clinical trial methodologists wrote commentaries on that. And basically, the first sentence of every single one was, well, you know, he's obviously right. I'm not going to talk about the paper. I'm going to talk about some of my additional ideas on this. So the reason why is, how many, can I ask here, how many people have been in a trial? You put up your hands, right? Out of you, how many of you have a formal training in trial design or statistics. Okay, if I did this in cancer, it would probably, so it was about 3/4 of you. In cancer, it would probably be about 10%, right? The people, how do people do their trials? They just copy everybody else. They don't really have formal training in it. And so if you just copy everything else, then everything sort of becomes forever conservative. So I'm saying in cancer, I'm a minority, a very tiny minority. In the clinical trial methodology community, I think most of the top clinical trialists would agree exactly with what I'm saying.
So can I ask one more question? Can we incorporate your suggestion into our guidelines? Is it possible?
Well, if it's not, why did you invite me? I mean, you know. Yeah, I mean, thanks for that, you know. That's funny. Fly down to Virginia and we'll ignore you. Yeah.
Oh. Hi. Yeah, that was a very provocative, interesting talk.
Yeah.
Can I ask, who pays for then drugs like BCDE that you were showing on your slides, since you said there was no cost?
Oh, well, the trial I'm doing is a surgical trial.
Okay.
So the patients are getting the surgery anyway, and we're comparing, you know, literally it's things like, do you sew up the incision vertically or horizontally?
Oh, okay.
So I'm not saying that, you know, oh, you can do all trials for free. Okay, I'm not saying that. I'm saying you can dramatically reduce the cost of trials if you do things like simplify down the inclusion and exclusion criteria, right? We use one. Right, I looked at some of the trials. Most of the criteria I saw in the trials that you sent to me struck me as completely unnecessary. Now, one example was that there was a whole list of neurological conditions that the patient couldn't have. So that means someone would have to check that the patient did. That makes, how many patients with ALS also have concurrent other conditions, and what difference would that really make to their forced vital capacity? Not very much. So that's the kind of thinking. How every time I'm going to make my trial a little bit more complicated, because in theory, maybe if there was a patient with Parkinson, in theory, you're making your trial more expensive, more difficult, and less patient-friendly. That's what I'm saying.
So, Dr. Vickers, I want you to understand this is an aggressive question.
Please, yes.
We have in the ALS field demonstrated benefit for patients taking part in clinical trials.
It's the same in every field.
Okay, so you're not asking us to throw away that benefit?
No, completely the opposite. Okay, studies have shown that patients do better when they're on clinical trials, and when no patients are on trials. And why are they not on trials is because we make it difficult for them. So why don't we make it easier for them to get on trials, get more patients on trials?
Sounds good.
So you're going to hear a little bit of the pessimism of a neurologist. I struggled with the concept of the adaptive design in this setting for two reasons. Then I've got a second question as well, if I'm allowed that. The reasons I've struggled with it are, number one, my suspicion of the pretest likelihood of success of a particular drug B, C, D, and E, is low.
Yeah.
And the effect size, even if one of them might be beneficial, is minuscule. So does that adaptive design process take, 50 years and 50 drugs? That's my first question. The second question is, riluzole is marginally beneficial. It's never saved a life as far as I'm aware. That said, do we run into issues with our IRB saying, we're going to randomize you to, drug B versus riluzole or drug C, D, and E, and if you're in arm 2, 3, 4, or 5, you will not be allowed to take riluzole, even though we're telling you it's the one drug that has benefit in ALS.
Okay, so, I'm not an ALS researcher, and I'm not going to put up on the board valid design. It's quite possible I'm put up on the board things that don't make sense, right? I just want you to take away the big picture. If you were going to do trials that were riluzole versus A and riluzole versus B, it would be better off to do a trial that was riluzole versus A, B, C, and D. You may not want to be doing that trial, but I'm sure whatever trials you are doing, you could rethink about in that format. Now, as in terms of adaptive randomization and effect sizes and so on and so forth, there's 2 points. You said pretest probability, or pretest likelihood. You can easily incorporate that in there. There's absolutely no problem. So you don't just say, oh, if you're better than riluzole, we'll increase your allocation probability. We can do that in a more sophisticated way, given the fact that you don't believe that drug is probably going to work. That's the first thing to say. The other thing in terms of effect size is, if you have the purpose of this design is, if there is a really good agent out there, this is the fastest way of finding it. Right.
Just time now for two short questions before we bring the other investigators up to public.
Really provocative talk. Just another question on the adaptive randomization. Have you had any experience with applying this on a multi-site research? Because with adaptive randomization, you're going to have a running process of evaluating each agent, right? And so
you're going to say, could you repeat that last?
So have you applied? What's your experience in applying this on a multi-site research level?
Okay. I personally haven't done this, right? Because I work in surgery, and this is drug therapy, right? But there are reports of this being done in the literature and in cancer, and I forget, there is a neurological condition that I think there might. I'm sorry. There is some, and there's one. I think it might be a muscular dystrophy in which, I may get this wrong, but the Central Muscular Dystrophy Society got tired of paying all the you go and do your phase 2 trial, and you go and do your phase 2 trial. They said, no, we'll take them out. You have a drug, great, right? We'll investigate it in a one phase 2 trial that we're going to be running centrally. So they do in prostate cancer. They do something. It's not quite the same as adaptive randomization, but it's something quite similar. It's not. People seem very fixed on that. It's just an idea. I think it's less important than the trial simplification, right? And making things patient friendly. I'd call it, it's sort of an icing on the cake. Focus on the cake, not the icing.
Thank you. It's a wonderful talk. Very interesting. And just one question, because we're all a little sometimes nervous about doing a comparative study, efficacy study, and having riluzole on the one arm and no riluzole or an experimental drug on the other arms. And I'll try to bring this into cancer. So let's say you have a limited small cell person and they want to do adjuvant chemo, and they choose, instead of the old cisplatin etoposide, whatever they were getting, the standard drugs versus other ones. Are there ethical issues in cancer research with doing this?
Well, yeah. I mean,
yes,
of course,
obviously.
But
yeah.
I mean, you deal with it on a case-by-case basis, right? And so, as I said, I wasn't trying to suggest that you start doing trials of riluzole versus A. I was just going to say, if this was the case, I mean, if I could say this was a placebo-controlled trial or whatever it was, I'm just saying, if that was, now it could be, you do riluzole, this is for people who stop responding to riluzole, right? And then, you either continue on it or you cross over to another drug or something like that. So, yeah, there are always ethical issues in any trial, and you have to deal with those on a case-by-case basis. But I'm saying, don't get caught up in the details. What's the big picture?
Okay, so that concludes the questions. Over here, Dr. Kieburtz and Dr. Beghi, we'd like the three speakers to come here, and my co-chair and I will go over the podium.
Well, I think let's open it up for another few minutes if there are questions for all the speakers and any of the issues, including the original ones we put up. I don't know if we can put that slide back. So any further questions to any of our speakers? One question here. Thank you very much.
Ryuichi from Japan, and I think this is going to be a question for all of you. I think in case of cancer, there are various kinds of stages. For example, very early stage and with metastasis, and various kinds of stages. And in case of other neurological diseases like CIDP, there are many subtypes responding to various kinds of different treatment. And for example, in Parkinson's disease, I'm also a member of MDS, but there are various kinds of juvenile Parkinsonism, Park 2, or just Park 8, quite different. And so I think it's right time for our ALS community to make a sensible subtype grouping depending upon the pathophysiology in time and clinical features. And so, for example, in advanced cases, there are a lot of neuroinflammation, and then we might be able to use anti-neuroinflammatory drugs. And in case of early stage, some other things. So I think I'd like to ask your opinion about that. In ALS, there has been no such definite subclinical classification or staging whatsoever. So most of the clinical trials fail, probably because SOD1 mice have the drug beforehand. And so that's a very different aspect of clinical trial and the animal experiments. And so I think I've read a kind of manuscript that that is going to be a very good classification of subtypes in ALS. So I do think it's the right time for us to think about that. So what about your opinions?
Okay. First, our speakers, but I know there are many other people here who are thinking hard about staging and phenotyping and how to do it. But comments, please.
Well, we discussed this issue several times today, and my impression is that we did not arrive to a conclusion, because we seem to face two opposite and two different situations. One is the idea to preserve the representativeness of the population that we are going to treat, in which case we should lump and have a huge number of patients in order to see the treatment effects comes out as a net effect, adjusting for a number of variables that represent the heterogeneity of the disease. On the other side, we have the need to look for very homogeneous population in order to identify what is related to the treatment effect by making the patients as homogeneous as possible, reducing at best the heterogeneity of the disease. But it seems from the discussion of today that we never came to a compromise on this. What I suggested in my presentation was at least to identify different time periods in the evolution of the disease, and then have separate trials in 3 different periods. It is early disease, the advanced disease but not in terminal phase, and end of phase disease, which represent 3 different situations. But I myself have problems in saying, knowing if I am correct in doing this, because even in these 3 large strata of the population that we are examining, we may have heterogeneity, in which case it's very difficult. I like, and I hope that tomorrow we will discuss pragmatic trials. And what I read in the handout was something that bothered me, because it seems that pragmatic trials are garbage. No, they are not garbage, because they are a good complement to regulatory trials, because they translate the efficacy of the treatment in the real practice, in which all the patients are treated and followed as individual practice. So I think that the picture is very complicated, but maybe that finding a compromise that on one side tries to identify the true treatment effect, and on the other side try to target all the patients giving the treatment. It's something that we can bring us to a solution of the problem, but I don't know if I'm on the right way.
Yeah. Well, in terms of heterogeneous, someone said to me before, cancer is not heterogeneous like ALS is. You have 5 people with cancer, you have 250 different types of cancer, because it's cancer cells that mutate, and you might have different clonalities and different lesions are actually different cancers. So it's a very heterogeneous, it's a very complex disease. I can just tell you some things that are happening recently. The drugs for cancer, cytotoxics, don't just kill all cells. They just kill cancer cells slightly more than they kill usual cells. And now we're moving more towards targeted agents. We understand a little bit more about that a certain pathway is upregulated in cancer, is downregulated, and we have a drug that would actually target that. There's been two different approaches. Sometimes those drugs are given, and the overall effects aren't very good. You have a small average effect, and the median survival increases by about 6 weeks. But you see in a small number of patients, about 10%, that the tumor shrink away and they live a very, very long time. And so once the trial is done, you've identified these exceptional responders, you try and work out what it was about those responders, and can you actually predict who's going to respond to your drug? The other thing is the new idea is what's called basket trials. And so it used to be, you have here's a trial for prostate cancer, and here's a trial for colon cancer, and here's a trial for breast cancer. Well, the new idea is, well, if we're targeting KRAS, it doesn't matter whether you're on KRAS prostate or KRAS colon, if you have a KRAS mutation and we have a drug that targets KRAS, we're going to test for KRAS and we're not really going to care about what the anatomic origin of your cancer. But that's all to do with the basic biology and a very clear mechanistic understanding of how the drug action, which we often do have in cancer, because of a very large number of patients and a lot of research dollars. I'm not sure you have that in your book.
There may be a middle ground, because as Andrew said, there are good examples in cancer, like with non-small cell lung cancer and gefitinib. There's very specific sensitizing mutations in the epidermal growth factor receptor. If you look at large populations, it looks like no effect or mild effect. If you look at those with mutations, there's a big effect. If you don't have mutations, there's no effect. We don't have that, but we could identify a priority. Say, rather than subsetting and saying this is the segment we want to pursue, in early phase trials or new phase trials, we could identify both predictive and prognostic markers, and the FDA speak, predictive being markers you think might confer sensitivity to the treatment, prognostic markers being about how you think the disease is going to progress differently, like young onset versus older onset. And then, as Andrew was talking about, response adaptive randomization is actually within a population start to see if those who have those prognostic or predictive markers are actually responding differently. And then starting to randomize those people with those markers preferentially. So it doesn't have to only, you can use response adaptation, not just on choosing drugs, but selecting population.
Okay,
yes. Identification of a segment that is biologically more likely to respond to drugs is fraught with difficulty. Yeah. Most of that is found empirically.
Yeah, but in the field of ALS, although I'm related to WFN research group myself, I hate to say that ALS Korea laboratory supported or some other things. So all lumped into that diagnostic category together. So I do think it's right time to just to find out subgroups, probably based upon the data from the failed clinical trials. For example, Dexpramexel study was reanalyzed by Hiroshi, and very, very good paper. And if you do reanalyze the failed paper, there should be a number of subgroups, which is responding, which is not responding. And then we might be able to find out responders. And just as Parkinson's disease, juvenile Parkinson's disease behave quite much differently from various kinds of diseases. PARK2, I mean, they have dystonia and other things. And so like that, my suggestion is learn from the failed clinical trials, just as Hiroshi did.
Yeah. Thank you very much. I mean, there is a problem about subgroup analysis that if you take one positive group, it follows logically that there must be a negative one, which you just tend to ignore. So I think that we have to be, and I'm sure we will come back to this issue of subgroup analysis as a way of defining significant groups is a very flawed way if it's post hoc. But and the other point I was thinking in terms of Andrew's points is that if you take a view that if we pursued this idea that we just get everybody in, what we would do then, instead of saying, let's be very selective and we'll wait, months before we find just the right patients for this group, we take them all in, and then when they're in, we then sort them into their phenotypic or their staging groups prior to, of course, reading out the outcome of the trial. So we don't bias that. But that may be a much better way than trying to limit recruitment, because we all know that the more you refine your inclusion and exclusion criteria, the less you recruit, as you were saying. Anyway, I'll say no more on that, because I think we'll come back to this discussion. There's a question here.
Yeah, the first two speakers, and this goes back to the comment that was just made, that we're starting to see more pharmacodynamic factors teased out of some of these basket trials. They did pretty well in the Cancer Research UK with Ian Walker in 5 or 6 lung cancer trials, or I'm sorry, 5 or 6 lung cancer existing products, and then the same thing with I-SPY 1 and 2, that didn't just rely on prognostic factors for the subgrouping of patients, but the differential response in the clinical experience of these patients in the different drug baskets. And I think that was a little bit different than the umbrella studies that had maybe one BRAF target, but several cancers. So this translates to my question in that the first two speakers discussed, I think these clinically observable traits that might be modeled as prognostic factors, which is very important, we talked about earlier today. But is there any experience outside of the cancer basket trials that we could perhaps lean on in neurodegenerative diseases of scenarios like that, of differential response to treatment in a particular basket that's been assigned to the drug, and then using that differential response as a predictive marker in a later stage trial?
In the field of epilepsy, unfortunately, at the present time, we don't have information that supports what we are looking for. That means looking for differences, for real differences between drugs that have been used for specific clinical conditions. Because if you compare these drugs given as add-on against placebo, you don't see any difference whatever drug you are using. The problem is that, unfortunately, many of the drugs that are used in epilepsy have been compared to placebo, and very few of them have been compared to active treatment on monotherapy, which may show you a difference in terms of seizure controls. But there is no interest on the industry to compare a drug that has been approved for use as add-on treatment, because there may be the risk that compared to another compound, it comes out to be worse. In which case, there are a lot of pressures not to do these drugs. And I'll tell you more. In the future, it's going to pass the idea that a drug for epilepsy could go into the market just after being evaluated as add-on without any need for use in monotherapy, and then the drug could be used in monotherapy in clinical practice. This is bad because obviously there is no way to have a direct comparison between these active principles to see if there is a real difference. Another possibility is to go back to the field that is in its infancy, that is to work on high-risk patients in order to prevent epileptogenesis. And in this case, we have some data from old drugs that again does not seem to work. That this is the reason why we say they are all symptomatic. But there is need to investigate new drugs to see if there is one of them or few of them that have an anti-epileptogenic action, in which case there may be differences. But unfortunately, we don't have such data.
Any other comments from the panel?
Sort of in a reverse way, in Alzheimer's disease, we know that people who don't have amyloid don't respond to anti-amyloid treatment. So that's a biomarker of treatment response. So, about a third of people in the Bateman trials didn't have amyloid accumulation. Those were negative trials, but if you look at the subpopulation, they appear to respond. The other is ApoE in Alzheimer's disease looks to be a prognostic marker. Whether it will be a predictive marker of treatment response is another question, but it certainly looks like they are more rapidly progressive and they may be more sensitive to other treatments, but their trials specifically ignoring those. So I don't know I can't think of a neurodegenerative disease treatment quickly that in which there is a biomarker which confers treatment sensitivity. Maybe somebody in the audience.
Thank you for that. I have a question for the panel. We have some evidence in ALS that early treatment may be more effective than treatment in more advanced disease states. And we've been hearing certainly some of the literature in Alzheimer's disease suggests that our clinical trials are coming in way too late when too many cells have already passed away. I wonder if in each of your disciplines there is a strong sense that early treatment is best, and is that influencing, therefore, your inclusion criteria for trials in your disciplines?
I can start for epilepsy because this is a field that I investigated personally. I am sufficiently old to have lived the period in which there was an enthusiasm on anticipating as much as possible the treatment in a person with epilepsy, to the point that it was suggested to treat the first seizure. And 20 years ago, there were a couple of studies. One was, I was very lucky to coordinate one of those studies, in which we randomized patients to be treated as a first seizure or to wait for the second seizure in order to be treated. And what we found out was that in the early follow-up, there was actually a difference because those who were treated at the first seizure had a relapse later on compared to the ones that were left untreated. But we followed this population for several years and we found out that in terms of remission seizure, which is much more important than just having a single seizure, there were no differences. So in epilepsy, early treatment, at least with these compounds, is working.
Thank you.
Yeah, I think Alzheimer's disease is trying to explain why the drugs fail other than they don't work. So hence going early must be the answer. I don't think there's any evidence that going earlier in Alzheimer's disease actually makes those drugs work better. It's a hope. There's some evidence, but not good evidence, not randomized. And in fact, there are not many approved treatments that only work when you go early. Even cholesterol reducing or HIV drugs work in advanced stages. It's hard to think of examples of drugs that fail to work entirely because the disease is too advanced. They may not work as robustly, but to find an absence of a treatment effect because the disease is so advanced is very rare in treatments. We also know from the HIV experience where very early on we treated early, the Concord trial. People actually had a worse outcome by being treated early because of the development of resistance. So it wasn't that there was a problem with that. It was a problem with how to use that. And now, of course, treatment is very early, but with multi-drug treatment. So I think the idea of going early as a solution is as many problems as going late. I don't think it solves problems.
It's not a law of the universe. It depends on the biology. I mean, treatment early is the whole deal in cancer. In solid tumors, if it's early, you cut it out and then you're cured, and that's it. That's the end of the story. And if it spreads, you revive your disease almost all the time. So what we try and do is find it early and cut it out, and early treatment works and late treatment doesn't. But that's because we understand cancer, we understand what a later stage means in terms of metastasis and in terms of local control. I think in some diseases, rheumatoid arthritis is an example, well, if you treat late, you're too late, you've already got damage. In schizophrenia, my understanding is you can treat as late as you want because, as schizophrenia as it progresses, it's not making any sort of permanent biological changes in the brain. So whether you treat early or late really depends on the biology of the disease and the mechanism of action of your therapy.
I wasn't talking about early treatment strategies are probably always more effective for patients than later treatment strategies, but the idea that drugs can't work because it's too late. That's actually great. Yeah, no, we are. Yeah, sorry.
Pat Andrus, question?
Hi, I can't help but to look at Dr. Vickers' pie chart up there and think that the 90% of our time in clinical research is spent on regulatory and compliance issues, and the 10% is spent on actually doing clinical research with the patients. And so I wonder, if this workshop will result in being more efficient about doing clinical trials. I think we tend to skimp on, we're so squeezed out by the compliance and the red tape. I think we skimp out on the outcome measures. I think we don't have much time to do things. And if we could do clinical trials more efficiently, we'd be in better shape. I've been doing this for 35 years, and the basic science has come light years ahead, but we're still in the same place clinically, doing clinical research as we were last, at the first Early House meeting. So I really hope that we can figure out a way to be more efficient about doing clinical trials.
Listen, I think you're absolutely right. There's got to be two sides to this, right? We need to attack the regulations. I mean, clearly clinical trials are ludicrously over-regulated. The classic example that's given amongst many clinical trialists and ethicists is that if I'm a doctor and you come to me with an infection, I can give you 1 or 2 antibiotics. I think, I'll give you this one. You go home. Now, if I then threaten to learn something, and I'm like, okay, I'll randomize you to these 2 antibiotics, then all of a sudden we're hearing about Tuskegee and the Belmont report and Auschwitz and all that kind of stuff. And there's this enormous weight of clinical trial regulation. It's really very problematic. We need to attack the regulatory structure of clinical trials, which has gone way too far. Okay. But at the same time, clinical trials bear a lot of responsibility because they bring a lot of it on themselves. And the example they already gave was inclusion-exclusion criteria. We have a regulation that we have to audit, we have to document, and we have to check that the patient really was eligible. And I think that is, in many cases, unwarranted, and we don't need to do that. But if it's there, we can save ourselves a lot of time and effort and focus on science rather than regulation if we cut down the number of inclusion-exclusion criteria to an absolute minimum that is necessary. And just for example, if we put in something saying that someone going on I saw it in one of the trials, someone on our ALS trial cannot be HIV positive. Right? Really, does that help the trial? Come on. I mean, excluding the one person, maybe out of 1,000 who was HIV positive, what effect would the HIV positivity have on the results of our trial? I don't know. But that means that every single one of those 1,000 patients, not only do they have to be documented as HIV seronegative, but then someone has to audit the fact that was done documenting those source documentation and compare that to the CRF. And you will avoid all of that regulation if you just remove that particular eligibility criteria. So yes, there's a regulatory burden. I'm saying trialists bring it on themselves by making their trials unnecessarily complicated.
Well, our panelists have brought to us some fresh perspectives from other fields in neurology and cancer and neurodegeneration, and we ignore these lessons at our own peril. We thank you very much, gentlemen.